SPFK, a clinical pharmacologist, Magister of Science, PhD. He is a PhD graduate from University of Groningen, concentrating in pharmacoeconomics. He is also a staff of Anatomy, Histology, and Pharmacology Department, Universitas Airlangga. And of course, our honored panelist, Dr. Asra Al-Fauzi, SAMM, SPBS consultant, FICS, FACS, IFAANS. He is a neurosurgeon working at Department, Faculty of Medicine, Airlangga, and Dr. Sutomo General Hospital. Interestingly, he is also majoring in economics and management. He is now the Chief of Faculty Office Ambassador, Faculty of Medicine Unair, the unit providing great contribution for this webinar to take place. Ladies and gentlemen, today we're having three discussion sessions that include presentation and discussion. Three fruitful topics. The first is drug development in infectious diseases, how drug targets, presented by Prof. Tomoyoshi Nozaki, MD, PhD from Japan. And Dr. Dwi Penny Kartikasari will serve as the panelist. Thereafter, clinical aspect and health problem of infectious diseases, multi-drug resistant bacteria will be delivered by Associate Professor Pinyo Ratanampawan, MD, MSc, PhD from Thailand. And Dr. Annette Arkom will serve as the panelist. And last but not least, health technology assessment of COVID-19 vaccine that will be presented by Prof. Dr. Martin J. Posma, PhD from Netherlands, with the panelists Asra Al-Fauzi and Dr. Abdul Khairul Rizki Purba. Each session for an hour, that is 45 minutes of presentation continued by discussion with our panelists. But participants are also invited to deliver questions to our speaker at the end of each session. Ladies and gentlemen, it's a privilege for me to introduce you to our first speaker, Prof. Tomoyoshi Nozaki, MD, PhD, Professor of Department of Biomedical Chemistry. He is currently the Vice Dean of Graduate School of Medicine, the University of Tokyo, one of the top Asian medical institutions. He is also Vice President of Federation of Microbiological Societies of Japan and the fourth President of Japanese Society of Parasitology. He graduated from Keio University School of Medicine and had postdoctoral training on molecular cytology, particularly genome plasticity, drug resistance of trypanosoma at National Institutes of Health, USA, and the Rockefeller University. After his return to Japan, he has been mainly working on virulence and metabolism of Entamoeba histolytica at Keio University, Gunma University, National Institute of Infectious Diseases, and the University of Tokyo. His current major interest in basic research include vesicular trafficking, thrombocytosis, phagocytosis, pathogenesis, organelle evolution, and drug resistance. He is working as editor at variable scientific journals. He is also vigorously involved in research and development of treatment against malaria and neglected tropical diseases. He is currently engaged with two drug development projects, one in the anti-leishmania screening and discovery, and the other in the anti-malarial hit-to-lead project. Therefore, he is the best person who can answer our question concerning drug development in infectious diseases, how to find the drug targets. Without further ado, please join us in welcoming Prof. Tomoyoshi Nozaki, MD, PhD.
Can you hear me all right?
Yes.
Yeah, okay, thank you. Thank you so much for a very kind introduction, Prof. Nurina. I don't know whether or not I'm a right person to ask a question about drug development in general, but surely that we have been working on the topic, and I would like to give you like two good examples showing our approaches trying to identify and develop the drugs and future drugs against common infectious diseases, in particular in tropical and subtropical countries. So I start my presentation after sharing my PowerPoint file. Let's see. Can you see my title slide all right?
Yes.
I don't know if it's appropriate to change the backgrounds of myself to this or to this, or if you want to stick to this, I'm all right with this, but maybe you can feel a bit more relaxed with
Which one is
the
comfortable for you to
picture
say?
from
It's okay.
Flores.
I said this is the, we just want to make a relax
I think
seminar,
he's
not
visiting Flores
No,
now.
no, my point is I want you to relax, not me to relax.
yeah, all of us should be relaxed, not particularly.
So I go with this then. Yeah. All right. So the next slide show the the content of my talk today. That I will start explaining the backgrounds of malaria and amoebiasis. I'm sure that you know that those tropical, I mean those the parasitic diseases, but I just want to touch upon the importance of drug developments against them. And I want to show you two examples for the drug developments using two approaches. One is the from natural bioresources, mainly like microbial resources such as fungi, actinomycetes. Second approach would be the drug discovery against malaria using a chemically defined compound library. So background of the malaria and amoebiasis. Of course, it's needless to say that the tropical infectious diseases, particularly parasitic diseases, are the global concern. Malaria, for instance, is endemic in close to the 100 countries, mainly in Africa, Asia, and Middle and South America. And it causes 3.4 billion risk of infections, over 200 million infected cases, causing to 400,000 deaths annually. And definitely this is the most important with the highest impact with the protozoan cause of mortality. And as you are aware, there is no established vaccine available. Recent development of RTS from GlaxoSmithKline showed some efficacy to lighten the severity of the disease, but it's very far from appropriate protection vaccine. And the treatment of malaria infection is mainly based on the artemisinin combined therapy called ACT against Plasmodium falciparum. And I think you're all aware drug resistance is a major concern for the treatment against malaria infection, not only for more severe form of malaria infection caused by P. falciparum, but also the infection caused by other Plasmodium species, mainly P. vivax in Asia. And both are artemisinin resistant and resistant strains of all available, clinically available compounds, as well as now clinically being developed, some candidate compounds which are developed and tested clinically. The drug resistance is so easy to develop in Plasmodium species. Therefore, constant refilling the pipeline of drug development against malaria is urgently needed and usually always needed. Amoebiasis is an intestinal infection caused by Entamoeba histolytica, and the over 50 million people infected, about 1% of total population on the planet, resulting in the death of 40 to 100,000 deaths annually. And this is a deadly protozoan infections important only after the malaria. No vaccine available, and metronidazole is the only clinically available drug, metronidazole or other nitroimidazole compounds, I mean. And as you know, that metronidazole is a tetragenic and causing severe side effects also. And resistance are very well reported in entamoeba, giardia, trichomonas. The latter two protozoan parasites share the common anaerobic metabolism together with Helicobacter pylori, which causes stomach cancer, stomach ulcer. Therefore, drug developments and identification of the targets, rational targets to develop, to initiate the studies leading us to the drug discovery against malaria and amoebiasis is urgently needed. Well, I want to emphasize the importance of partnership for the drug discovery and development because I'm just a parasitologist with a medical degree, and I don't have a very good background as a chemistry, safety testing, animal testing, etc. So the partnership with my with a variety of disciplines, including experts on product development, such as pharmaceutical industries, also the product development partners, such as the Drug for Neglected Drug Disease Initiative, DNDI, as well as MMB, Medicines for Malaria Ventures, and also TB Alliance, Global Alliance of Tuberculosis, for tuberculosis. And I want to give you two examples. One is the bi or trilateral international collaboration with the Indonesian counterparts. The project was supported by AMED, Japan Agency for Medical Research and Development, and also JST, Japan Science and Technology Agency, together with JICA, Japan International Cooperation Agency. And this is the drug development using microbial resources as a source of the compound. This is against malaria and amoebiasis. The second example shown in this slide is called private-public partnership or public-private partnership, PPP. This is through collaboration between government, academia, and also pharmaceutical industries, as well as product development partner, abbreviated as PDP. And in this specific case, I want to thank Medicines for Malaria Venture, MMB, for their continuous support. And this project has been and will be supported by a big funder located in Japan's office, a Global Health Innovative Technology Fund, GHIT Fund. I will start with the first case, which is international collaboration with Indonesian fellows. Indonesia, despite the big impact of infectious diseases, particularly parasitic diseases, drug developments against such tropical diseases, the materials for the drug pharmaceutical industries are largely limited. And I heard from my colleagues in Indonesia that materials needed to produce the drugs, 90%, more than 90% of drugs used in the countries are transported or imported from overseas. And there is no domestic capacity to develop new drugs, whereas while that you have a capacity to make a generic compounds. And the as I said, artemisinin combined therapy, ACT, is the standard chemotherapy against malaria. However, that there is very low availability of standard antimalarial chemotherapies due to a lack of sufficient reagents and compounds. So we Japan has a strong background and expertise on the drug developments from microbial resources. If I'm allowed to take one example, Professor Satoshi Omura from Kitasato University, who is a Nobel laureate of the physiology and medicine 5 years ago, who discovered and developed anti-filarial, anti-onchocerciasis drugs called ivermectins, and he derivatives it into ivermectins, which is used and donated to treat in Africa and Asia and South America to treat onchocerciasis and filarial patients for free. And Indonesia, on the other side of it, is a very rich in microbial resources. Actually, Indonesia is the second largest mega biodiversity countries thanks to a big land area, a variety of geographic features, and many islands. Actually. This is only after Brazil in terms of mega biodiversity. So taking advantage of those two strong points, I'm proposed about 7 years ago to JICA and AMED. We would like to initiate this collaboration to create a new platform for drug development. So this project was called SATREPS, Sustainable Developments of the Science and Technology from AMED and JICA. And with Indonesian collaborators, we declared we will be searching for lead compounds against malaria and amoebiasis from microbial resources. So in this collaboration, which we initiated 7 years ago, we asked Biotech Center of BPPT, the Agency for Assessment of Application Technology, to take an initiative. And with Airlangga University, ITD, Institute of Tropical Disease, as well as LIPI, we initiated this project. And again, we took advantage of mega biodiversity of microbes as well as plants, and try to use them as a source of the interesting novel compounds, which can be further used downstreams for chemotherapy and prophylaxis against malaria and amoebiasis. Oh, by the way, in case that you're wondering about what SATREPS means, this is a rather complicated abbreviation such as a complicated name, science and technology, research, partnership for sustainable developments. So this really strongly matches the common agenda of SDGs in coming 10 years. So I'm sorry that somehow the transition of the slides is rather slow. I really don't know why.
We can wait, Prof. It's okay.
We can wait.
Can wait.
Right. Yes, right. So this is how we do the research. So aim of the projects is again identify microbes and compounds which inhibits specific and exclusive enzymes present in Plasmodium falciparum and Entamoeba histolytica by screening Indonesian microbial culture corrections, and further purify the active compounds and determine their structures, and finally testing them for the efficacy and safety using rodent models, and finally claim and propose a lead compound for the downstream clinical preclinical developments. And methods of protocols very briefly, if you're interested in particular assay, I'm happy to share that. Although at the beginning, we targeted specific enzymes such as mitochondrial respiratory enzymes listed in the middle, p-pharocyparum dehydroorotate dehydrogenase, malate quinone, oxidoreductase, NADH dehydrogenase, and the phospho coenzyme A kinase from Plasmodium falciparum, and cysteine synthase and serine acetyltransferase, NADP oxidoreductase, not kinase from Entamia histolytica. I'm not gonna go into details to explain the targets. And again, if you're particularly interested in why we highlight those mitochondrial and the amino acid metabolizing enzymes, I can come back to you later with the questions Q&A session, or if you can send me a letter or mail that I can respond to you accordingly. Right, and we also not so we started enzymological screening using those enzymes listed here as a targets. We concurrently that we also tested culture extracts against cell culture. So for Plasmodium falciparum, we use the in vitro culture using human erythrocytes. For Entamoeba histolytica, we used arsenic and the anaerobic culture of Entamoeba on 96 well plates. Then expected outcomes when we started this project. So on our side, we expected that we can get access to very rich, abundant Indonesian bioresources for future drug development. Such important libraries and the resources can be expandable to drug discovery against other non-communicable diseases. And our government, Japanese government, emphasized the importance of medicine-driven economical developments. And this is a part of Fifth Science and Technology Basic Plan. So this really benefits us on Japanese side. On this other side of it, for the Indonesian part, this project will help the establishment of infrastructure of the drug discovery. And it may also enhance the possibility of the domestic production of drugs against a variety of diseases, again, not only infectious diseases, but also non-communicable diseases. And this also fits the master plan for acceleration of Indonesian economy developments 2011 to 2025, MP3-1. Right, so the first phase of the project is terminated at last March last year. So we have started second phase of projects, 6-year projects next April, last April, sorry, April 2020. Right, so as I explained that the Indonesian part partners are mainly Biotech Center, BPPT, Airlangga University, ITD, Indonesian Institute of Science, LIPI. And on Japanese side, we have four members, U Tokyo, myself, Kitasato University, Nagasaki University, and a venture company for the pharmaceuticals, Microbiopharm Japan. So to make a very long story short, I just explained major outcome of our research project. So I start with discovery of anti-amoebic compound. So we screened about 7,000 microbial extracts and identified approximately 20 to 30 extracts showing a sufficient in vitro killing activity, lacking a toxicity against human cells in counter assay. When I say it, counter assay, this is for the exclusion of toxicity against human cells. So out of these 22 fungal extracts, we identified very interesting compounds. So those compounds are shown here, fumagillin and ovallicin. They share the common structure, as you might be able to identify. It has the cyclohexane with spiral epoxy side sticking out of it. So this is a spiral epoxide cyclohexane as a core structure. And actually, this spiral epoxide moiety is the functional unit. So ovallicin is a very simple structure with the isoplanoid-like residues as a functional group sticking out of these positions, whereas fumagillin has the also additional moiety sticking out of it. But we found in the current projects we are working on right now that this moiety is really not needed for the activity. Fumagillin and ovallicin are very well-known angiogenesis inhibitors. This was actually identified not by us for the first time, but this was identified about 60 years ago as anti-amoebic compound already, 60 years ago. And as a matter of fact, this was also clinically tested against amoebiasis patients. Fumagillin showed moderate efficacy to treat amoebiasis patients and very little toxicity, well, acceptable toxicity against in human patients. Remember, this is 60 years ago, so there is not much ethical clearance needed at this stage. So there is no clinical study, no safety testing, nothing. All right, at the same time, ovallicin has never been tested against humans, of course, and against amoebiasis properly. So once we identify those two compounds from Indonesian microbial resources, we tested them in vivo using rodent model. Before moving to in vivo testing, I simply want to show how competent, how potent those compounds are compared to metronidazole, which is a clinically used compound against human infections. So this is a structure of metronidazole, which is a 5-nitro imidazole compound, which shows the IC50 or EC50 efficacy concentrations of 50% inhibition, growth inhibition, about 2.5 micromolar. Okay, and if we compare IC50 of ovallicin and fumagillin, this is about 20 to 70 nanomolar. So this is about 100-fold less in concentrations. So 100 times more efficacious against in vitro culture of Entamoeba histolytica trophozoites. Okay, and importantly, neither ovallicin and fumagillin show toxicity against human non-cancerous cells. In this case, fibroblast cells, MRC5. So IC50 value is about 26,000 nanogram per ml. So selectivity index, safety index is over 4,000 for ovallicin, more than close to 100 for fumagillin. So we can conclude that ovallicin and fumagillin are very efficacious, as well as very safe compounds against amoebiasis. Right, so we also tested in vivo. So we can take advantage of the liver abscess models using hamsters. So we directly inoculate amoebic trophozoites into left robe of the hamsters, and then start treatments on the second day for 4 times. And after the 5 days, we sacrifice animal and weigh abscesses and show the efficacy as the percentage inhibition of the abscess size compared to the entire weight of the normal liver. So unfortunately, and this is due to the different metabolisms between human and rodents, ovallicins and humagirins are not orally active. In other words, this is not efficacious to kill the parasites when administered orally. Instead, when ovallicin is directly subjected via a subcutaneous route, this is amazingly efficacious. About 1 milligram per kilo of the ovallicins cured almost 100% of the amoeba-caused liver abscesses. And this is about 10 times better, 10 times lower in dose compared to metronidazole when administered orally. This lack of the oral availability is due to the differences in P450, cytochrome P450. And this is not the case for human enzymes. The target of the humagirin or ovallicin has been already demonstrated. This is the translations of Entamoeba histolytica. The methionine aminopeptidase 2, MET-AP2, is known to be a target. And MET-AP2 cleaves off the methionine in the nascent polypeptide chain. And this is, for that reason, this is essential enzyme for the parasite. And human has a 2 aminopeptidase, MET-AP1 and MET-AP2. And this can be the reason why ovallicin and humagirin, which inhibits MET-AP2 only, but not AP1, inhibits the proliferation of Entamoeba histolytica and cured amoebiasis, but it's not toxic to humans. Right. We also discovered other anti-proliferative compounds from Indonesian bioresources. I'm sorry, I think my PowerPoint is crashed. Let me open it again. So this is the anti-amoebiasis drug development part. Right. Yes. So the next I move to anti-malarial drug discovery. Again, to make a very long story short, we identified a very interesting and potent compound called borelidine. Borelidine is a macrolide antibiotics first isolated from Streptomyces rothschildi. And this was named as borelidine after the activity against Borrelia. And this is the macrolide cyclic compounds. Okay. And again, the target is known. It selectively inhibits threonine tRNA synthetase again. And this is against inhibits translational machinery. You may be aware that the other amino acryl tRNA synthesis AARS inhibitors such as mupirocin, harafuginone, and clindamycins. Those inhibitors are very well developed, targeting a variety of tRNA synthetase. in cancer cells as well as infectious agents. So again, tRNA synthetase of carrying a variety of amino acid anticodons are very well established targets for drug development. And again, I'm showing small here that IC50 value against p-falciparum. This is less than 2 nanomolar. So, and this is much more efficacious against most, or I would say all of the antimalarial compounds used clinically right now, including, of course, in chloroquines and quinines, mefloquines, and anti-sulfur compounds like SP, PD-mesamine, sulfadoxine, as well as artemisinin. So again, this is very potent, but unfortunately that the this borodin compound is also slightly toxic to the human cell. So we need a further drug modification to increase efficacy as well as loosen its toxicity to human cells. For a discovery against inhibitors of malarial enzymes, as I said, we took like 3 major candidates such as DHODH, dihydroorotate dehydrogenase, malate quinone oxidoreductase, as well as NDH2, NADH oxidoreductase 2, all of which are involved in energy production in mitochondria. The last 2 are involved in electron transport chain, whereas DHODH is needed for pyrimidine biogenesis. And DHODH particularly is a very well established drug targets against malaria. And a few compounds already, including DSM-265, developed by Takeda, has been entering into the phase 2, phase 3 clinical trial right now. And other than that, there are multiple candidates which are known to inhibit DHODH in the pipeline of drug development and antimalarial. Other than borodin, we also identify about 10 compounds listed here from I show in a couple of a few slides shown here. And good news is we identify so many interesting lead compounds. The bad news is all of those compounds are previously identified, and some activity against infectious agents, not necessarily against malaria, have been demonstrated. So structure-wise, scaffold-wise, they are not a novel compound, except for the one compound we identified as a cyclic peptide, which we cannot show here for the patent issue. But other than that, all of those compounds are structurally known in the past. Okay, with this, so this slide illustrates how many, a variety of the compounds with a different structure we identify from natural resources in the Indonesia. So basically, during the project, we sent experts on the microbes to like a variety of islands in Indonesia, and ask our Indonesian colleagues to collect samples from soil and plant litters, and to isolate fungi and actinomycetes. And those microbes are stored at Biotech Center BPPT, as well as INSCC, Indonesian Culture Collections, LIPI. So hopefully the important microbial resources will be available for Indonesian researchers who are interested in drug development or use of those precious bioresources from Indonesia in the future use, for the future use. All right, so to summarize this part of my talk, we identified several major lead compounds that have a potential use for future developments as anti-amoebic and anti-malarial drugs by a bilateral cooperation, a SATREPS program. This program combined strong points and advantages of the two countries. And through this project's microbe library of more than 20,000 was constructed from Indonesian bioresources and utilized for screening and should be available for open use in incoming future. And technology needed to microbial collections identification, archiving, cell and enzyme-based screening, and structural elucidation were transferred to Indonesian researchers during this project. And we have proposed, we proposed actually 2 lead compounds against amoebiasis and 1 compound against malaria for next stage of developments. Obalacin humagilin for as anti-amoebiasis compounds and boreridin as anti-malarial compounds. So what next? So we propose to JICA and AMED again for the continuation of the projects, and we are lucky enough the project and our proposal was funded last year. So this was officially initiated from April 2020, and second year has been just started a few months ago. Okay. I think you might be wondering what I'm holding it now, but I can show you that. Yes. Yes. All right. Yeah. So the project has officially started. So in this second phase of project, which will continue until 2025. We are going to take the lead compounds against malaria and amoebiasis for preclinical testing. And so combining
Prof. Zaki, it seems that Prof are having connection problem. Prof. Nozaki? Yeah, we still wait. Yeah.
Maybe we can go to Dr. Penny, maybe?
I would like to summarize, yeah, a little for what Professor Masaki has been present. So in the drug development, since all of us has a specific knowledge and skill, so it's very important to build a partnership with multi expertise, yeah, such as with the biochemistries, pharmacologies, maybe microbiologists and parasitologists. And then we should collaborate. So this is very important in drug development. What Professor Nozaki showed us is how Professor Nozaki and his team started this project in anti-malaria and anti-amoebiasis from 5 years ago. And other things that he's highlighted is how to find it. First is with the screening.
Prof is coming out.
Oh, okay. Hello, Sensei. We lost you for a few minutes.
Prof,
can you hear us?
How?
Oh, it's not clear, Prof. We can hear your.
Sorry. How long? I'm lost.
Oh, it's only a minute, I think. And
it's all right.
Dr. Penny.
Okay.
So now you're seeing my slide all right?
No, we're seeing you.
We haven't. We're still
No.
waiting.
I see.
Okay. Thank you.
All right, so this is the collaboration scheme of the second phase of the project. University of Tokyo and Nagoya Institute of Technology is responsible for chemical synthesis and chemical structure modifications, optimization. Bozo Research Center has expertise of safety testing, animal testing, and this is the CRO, clinical research organizations. So they are experts for drug developments. And this time we started a trilateral collaboration instead of bilateral. So Indonesian partners as well as Malaysian partners who have expertise in the chemical synthesis are also a part of the team. Primate Research Center, IPB University, plays a major role in the animal testing, particularly like a non-human primate model of the malaria, amoebiasis, tuberculosis, and dengue. Right. So second part of the my talk is a public partnership. And this is through the funding from GHIT Fund, Global Health Innovative Technology Fund, with a collaboration with the famous, very renowned product development partner, Medicines for Malaria Venture. Right. So this time we started this project about 3 years ago, and the first phase is each phase consists of only 2 years. We are entering the second phase of 2-year project right now. So instead of taking advantage of the natural microbes as a source of compound, this time we took advantage of a compound library of known structures. So we used compound library from Drug Discovery Initiative, University of Tokyo. And this is a compound panel of the about 200,000, sorry. 200,000 compounds of validated structures as well as the fragment and scaffold library. And again, this time not a 96 well plate based, but we used either a 384 well plate or 1,536 well plate that we accomplish high throughput screening against malaria culture. And after profiling the hits and hit deconvolutions and safety prediction and testing, we narrow down the candidates to the level that we can propose a few series of structurally novel compounds for the further derivatizations for lead optimizations. So, right, I see something. Okay, so this is how we did the experiments. And out of 210,000 compounds, so we identified approximately 1,000 hits, which after the primary screening with the a threshold and hit criteria of 90% inhibitions at a single fixed concentration of 2 micromolar after 72 hours of incubations. And after the confirmation of the hits using 2 different bioassays using lactate dehydrogenase assay as well as a cyber-green assay, which the second one, cyber-green assay, is to stain DNA to see the decompositions of the genetic DNA, genomic DNA. So we narrow down the hit to about 600. And also, since, as I said, DHLDH has been seen as the rational drug targets, and there are quite many compounds which inhibits DHODH in pipeline. So we avoid trying to get the compounds targeting the DHODH. So we screened against identifying inhibitors against DHODH by screening against a 3D7 P. falciparum standard strain 3D7, which it transforms to express exogenous yeast-derived DHODH. Okay, and further down, that we also tested a DD2 strain, which is chloroquine and also SP resistant cell line. So we narrow down to about 500 hits. And further, we determined IC50 and try to prioritize the hits. And the MMB also applied multi-parameter optimizations. So they took into account like a potency, physical chemical property, and safety index, and structure alerts. And they also have the libraries, a database such as MMB database, as well as Chembo database. So they basically want a novel structures showing antimalarial efficacy. And finally, the chemists, we call it chemist I. So 3 directors of MMV, which are chemists, took look into the chemical structures of those 500 compounds and just handpicked like a 3 series of the novel structure for the further developments. So actually we are...
Excuse me, Prof. Excuse
yes?
me.
You lost me
I beg
again?
your pardon? No, it's still okay, but would you please switch the presentation to the presentation view? Because
Oh, this is not presentation view yet?
yeah, yes.
Oh.
Would you please?
It's okay now?
Haven't been seen.
What do you see now?
We see nothing.
You see nothing.
Yeah, it's okay. We can wait. Oh yeah, it's.
When did it start?
I think this slide.
This is full view?
Right. Yeah.
Okay. So from now on, we are going to produce derivatives and series of derivatives of the 3 series, which I cannot show at this moment. And we are going to check the life stage activity, and in other words, whether or not those compounds are efficacious against erythrocytic stage or sexual stage, mosquito stage, as well as liver stages. We are also carefully monitoring rate of kill, how quickly the compounds kill the parasite in erythrocytes. Rate of kill is quite important because we want a cidal compounds, but not a static compound. And we are also analyzing MOA, mechanism of action, which is quite important. Try to avoid the overlapping mechanisms. As I explained, that we don't want any more inhibitors targeting DHODH for the same reason. We really don't want to target other mitochondrial enzymes such as NDH2 or complex 3. And again, like ATP4 and also CAR and the PI4 kinase, those are very well known targets to develop antimalarials. So we also want to avoid those. So how do we study and identify the mechanism of action. The routine way of we are doing it is we maintain the 3D7 parental strain with increasing concentration of drug, finally to yield a resistant cell line, hoping that some important mutations which is responsible, which may tell us the hints for the drug targets, uh, uh, the gene responsible for uh, uh, the drug mechanisms mutated. And this is a very common way that the malarial researchers identify the targets of the known or the drugs under developments. And then further down, edema, absorption, desorption, metabolism, excretion study will be done. And oral bioavailability is quite important to ensure. So going through that, and we are also do administer cytotoxicity assay and the cardiotoxicity assay. We are also testing the compounds against a panel of resistant strains, including artemisinin-resistant cell line. And this is the way that we are going to do in the future. So this is the screen prioritization scheme that I just explained to you already. And out of those 4 compounds that we are going to synthesize, derivatize the related compounds. Right, so the unfortunately a new proposal for hit to lead optimization, hit to lead development has been funded by GHIT Fund for coming 2 years. So the 3 new series will be structurally optimized for better physical chemical properties, drug-like properties, and for a better improved efficacy and acceptable toxicity. And of course, the lead compounds must have the appropriate pharmacokinetics and pharmacodynamics properties. And of course, they must be safe for human use. So with this, I hope I was able to show you like 2 cases of this partially successful at this stage, but hopeful that the incoming years for leading to giving rise to the propositions of the lead compound for the clinical testing in coming years. And I think such study cannot be done without collaboration with many collaborators. Particularly, I want to thank our collaborators at Airlangga University in these occasions who are involved in the anti-amoebic drug developments like Mirna, Adianti, Lydia, Tumeu, and the Ilmi. And also this is done at the Institute of the Tropical Disease headed by Maria Inge Lucida with the help of Ajmal Fuad and Ati Vidi Warusayanti and many other people who help us with that. And also with the collaboration with TB Alliance, Professor Madi's group in medicine also supported us for anti-TB drug developments. So Rebecca and Debbie have been helping us for anti-TB screening, and I'm very thankful to them. And we have quite many Indonesian students in our master and PhD courses, including Yulia, Farida, Devi, and Defi. And also Penny is currently my PhD student who is finishing her PhD with me. So with that, again, I would like to thank other Indonesian partner from LIPI and PSSP, IPB University, as well as BPPT. And also I want to thank our collaborators who kindly supported us during anti-TB drug developments, Professor Shimizu and Professor Utsumi, and also Professor Mori from Kobe University, who are generous enough to let us use a PS3 facility at ITD. With this, I want to close my presentations. And I'm happy to take any questions if you have any. Thank you for your attention.
Thank you, Prof. Mazaki, for the impressive presentations. You remind us again about our potential of having mega microbial diversity that can help us addressing the problem of antimicrobial, anti-malarial resistance. And we're now entering discussion session. First, I'd like to invite Dr. Dwi Penny Kartikasari to provide any comments or question to Prof. Mazaki.
Yes, Sensei, this is very interesting to hear your presentation now. This is very simple questions from many kinds of resources. Maybe the attendant here wants to know why you choose microbial resources as the drug, for the drug compound. Because usually people here using from plant. Mostly we are using from plant like leaves, bark, and etc.
Yeah, I think basically we want to avoid repetitions because I know that the Indonesia has a very good history of the dealing with like Chinese medicines and like
traditional
jamu. So I think quite many studies have been done trying to identify the drugs against infectious diseases. So jamu is a very good way of doing it. We don't necessarily exclude the possibilities of taking plant extracts as a source of screening. So it's also include, actually we did it actually. So, but then we didn't really identify anything interesting from plants at this stage.
Are there any, difference between, if we're using microbial, then we need other techniques to do it, like we have to culture it or something like that.
Yes, that's right. So again, we want to take a strong point from Japanese side and the Kitasato University and also Institute of Microbial, microbial chemistry, Bikaken. It has a very strong backgrounds as a to provide microbial resources. So basically, we want to bring into such expertise into Indonesia. So this is the only reason. And once we establish stock in the refrigerator freezer, then they can be regrown, put it back to the ambient temperature, and again, such resources stored can be used for a long time. Also, replenishment of the library is always needed. But again, it's better than the plant extracts that we have to hunt for plants every time you need it. And by the time you want the plants again, maybe plants are no longer growing there. So unless a plantation is established, then such drug discovery can be suddenly interfered. So for that sense, microbes are more convenient and reasonable to correct.
Are there any specific microbial that we should use, like only actinomycetes or fungi or?
Yes, I'm not the expert of microbes myself. I'm expert of parasites as you are. So we just simply follow that our instructions and preference by Kitasato people who have expertise in actinomycetes and fungi. So that's only to measure.
Since those are already established in Kitasato University, and as you told us before, that partnership is very important. So that's why, yeah.
Yeah, thank you for reminding that again.
Yeah.
That the partnership is important. I cannot do anything without the help from such experts.
Yeah, and then for the finding the enzyme, drug target, what is the key point to find? This is very important.
I think that's the title of my presentation, right? I was supposed to speak on that, but I just simply skipped it because it's such a difficult topic. For instance, such as like anti-tuberculosis drug developments and anti-dengue drug developments, that there have been many studies targeting specific unique enzyme present only in tuberculosis, on in MTB, for instance. But most of those studies have been miserably failed. This illustrates and underpins the difficulties of drug developments against tuberculosis, for instance. For some reason, all the compounds which inhibits enzymes present uniquely in the pathogens don't really effectively kill the bugs, bacteria, viruses, or parasites. And again, drug needs to be taken up and needs to be metabolized properly, but not degraded. So the drug needs to reach to the targets, and this is really a complicated issue. That's why all the big pharmaceutical companies are now shifting like a target-based drug discovery toward like a cell-based screening. So we just come to like a structure-based studies and target-based studies, and finally coming back to the cell-based screening. So now that I know that, for instance, Pfizer and Johnson Johnson has been developing anti-dengue compounds, and I think they are in the phase 2 and 3 clinical trials. But those lead compounds are discovered through, again, the compound libraries, screening of compound libraries using cell-based screening, but not the target-based screening. So I don't know. Identifying unique targets is easy. And we did it. So like a DHODH, MQO from malaria, and cysteine synthase, NAD kinase from entamoeba, and protease, serine protease from dengue, and coenzyme A biosynthesis from MTB, M tuberculosis. We have been doing it for years. But again, the questions, have we found any good leads which can be taken to clinical testing? The answer is no. So again, we are sort of, back and forth, coming back and forth, whether which is a better way, either a target-based drug discovery is a faster way, or cell-based assays, after all, is the best solution.
Yeah.
Okay, Dr. Penny and Prof. Nozaki, I think it's time for us to read one or two questions. The first is from Muhammad Kanedi. I think he's just want to confirm the problem we have about anti-malaria resistance. He said that the result of a study by researcher at the University of Lampung showed that malaria falciparum was resistant to artemisinin. So it seems to confirm how important your work for the development of the promising new anti-malarial drug. And the second question is from Abdul Karim Rizky Purba. He asked about, I think, a similar question here with Dr. Dwi Penny. It's the first, do you have any suggestions which bacterial or fungal can we choose? And the second, do we need to have special concern about the passage of the bacterial culture? Can you catch the question?
Yes, I did. Thank you for asking questions. Artemisinin resistance is everywhere now and spreading throughout Asia also. So you confirm artemisinin resistance in Indonesia? That what he or she said?
Yes.
I see. Okay, which I wasn't aware. So thank you for the information. I catch up with that. The passage of the microbes are quite important. We experience many times for the first screening, some microbes is effective. And after making again the extracts and culture again for the second time for reconfirmations, sometimes we lost the activity. And we apparently don't have a good explanation for that. So again, passage of microbes are quite important. And this is where the microbiologists and experts on the microbes, the fungi or actinomyces are really needed for sustainable maintenance, long-term maintenance and archiving of your microbes. And I think it's a key question. And we have experts who can support you with that. But without the expert support, it's very difficult to sustain, maintain the good quality of the microbes. That's quite true. It's very tricky. And don't ask me how, because I'm not expert on that, but I can connect you or whoever who are interested in with appropriate person.
Thank you, Prof. Nazaki. Thank you, Dr. Dipani. It's such a great discussion, but due to limited time, we need to end this first session. And I would like to take this opportunity to extend my sincere gratitude to Prof. Nazaki. Thank you for taking your time here.
Thank you very much for your attention and question. Bye.
Ladies and gentlemen, now we are in the beginning of the second session, and I'm so delighted to introduce our second speaker, Pinyo Ratanampawan, MD, MSCE, PhD, C.Med. She's from Thailand, our neighbor country that is leading in research of infectious diseases and tropical medicine. She is currently an associate professor of medicine at Division of Infectious Diseases and Tropical Medicine, Department of Medicine, Faculty of Medicine, Siraj Hospital, University, Bangkok. Prof. Pinyo obtained MD degree from Chulalongkorn University, thereafter taking graduate diploma in clinical science and residency in internal medicine from Chiang Mai University. Then 2 years fellowship in infectious diseases at Siraj Hospital. Afterward, she pursued master and PhD studies in epidemiology at University of Pennsylvania, USA, focusing in pharmacoepidemiology. She also took variable courses in infectious diseases and tropical medicine, and also obtained certificate in principles of clinical pharmacology from NIH Clinical Center, USA. Her long list of publication truly signify her intense work in research of antimicrobial resistance. Therefore, I believe she will provide clear explanation about clinical aspect and health problem of infectious multi-drug resistant bacteria. Without further ado, please join us in welcoming Prof. Pinyo Ratanampawan. Prof. Pinyo, you have 45 minutes for presentation.
Okay, thank you so much for the great introduction, Dr. Nurina. May I share my screen? Okay. So it's a little bit delay. Okay.
It's okay. We can wait.
Okay. Okay. So thank you so much for the meeting committee for inviting me to join your webinar for the second time. Today, the title of my talk is Clinical Aspects and Health Problem in Infectious Diseases, focuses on multi-drug resistance bacteria. And because my main work is a clinician, I will focus more on the clinical cause and therapy. And this slide shows my disclosure over the past 5 years, research grant, honorarium, and travel grant. Okay, today's topic of my talk will be categorized into 2 groups. The first one, I will talk about global burden and epidemiology of multi-drug resistant or MDR bacteria. And the second part will focus on therapeutic option for MDR bacteria, either old agent and also novel agents. So let's start with the first part, global burden and epidemiology of MDR bacteria. And I'm not sure if anyone can guess who is he. Yeah, it's kind of difficult to do an interactive lecture because we are on Zoom right now. His name is Charles Darwin, who is the owner of Darwin's theory. He said individuals with less adaptive trait will less frequently survive to pass them on. That's why right now we see the emergence of bacterial resistance ongoing as ongoing problem, because only the drug-resistant bacteria can survive in the antibiotic era. So this is the Charles Darwin and theory of evolution. And this one may be the familiar face for the pharmacist, I guess. His name is Sir Alexander Fleming, who discovered of penicillin nearly 100 years ago. And he accidentally discovered penicillin in the bacterial culture plate. This slide shows that this is a Staph aureus colony, and penicillium colony spill on his plate, and penicillium creates some enzyme and some agent that can inhibit growth of Staph aureus in the plate of bacteria. So this is the first time that he discovered penicillin, and he is the father of antibiotic at the moment. This slide shows the timeline of antimicrobial resistance. As you see now, in 1940, penicillin was discovered. Few years later, Staph aureus developed resistance to penicillin. And in 1960, we discovered methicillin. And few years later, Staph aureus became resistant to methicillin. Ceftriaxone, which is the well-known drug in the moment, was developed in 1980, and few years later, the bacteria start to create ESBL, which is stand for extended spectrum beta-lactamase enzyme. ESBL producing bacteria are very resistant to ceftriaxone. And imipenem, which is the first carbapenem in the world, was developed in 1985, and few years after that, many bacteria can produce carbapenemase, naming MC, KPC, or Klebsiella pneumoniae carbapenemase, or NDM, stand for New Delhi methyl-beta-lactamase enzyme. That's why new antibiotic cannot last long in the market. The problem of bacterial resistance is not just the Asia or Thailand problem, it's also the world problem. Everyone faces the problem of bacterial resistance. We have many data from all across the world, but for Thailand, it is estimated that there are approximately 140,000 episodes of infection caused by resistant bacteria. And it is estimated that there are approximately 30,000 deaths per year, yeah. And at least $2 billion USD loss in productivity every year. So that is a lot burden for the lower middle income country as Thailand. So another term that we usually use, AMR, is stand for antimicrobial resistance. It is considered as one of top 10 global public health threats by WHO. In addition to COVID-19 right now, okay? And for the main driver that can cause AMR, including misuse and overuse of antibiotic, lack of clean water and sanitation, and also inadequate infection prevention control. And Global Antibiotic Research and Development Partnership, or GARDP, aims to develop at least 5 new treatments to target MDR bacteria by WHO by 2025. But right now it's 2021, so we have only 4 years to develop 5 new promising agents to fight against MDR bacteria. To be on the same page, I would like to be sure that everyone know the definition of drug resistant. For multi-drug resistant or MDR is defined as resistant to at least one drug from at least 3 antimicrobial categories. But for extensive drug resistant or XDR, the bacteria must susceptible to more than one drug from less than 2 antimicrobial categories. And the last one is pan-drug resistant or PDR. It means the bacteria are resistant to all antimicrobial agents that are available in the market. So we have MDR, XDR, and PDR. Many abbreviations. Okay, and this slide shows WHO priority pathogen list. There are 4 pathogens in the critical priority list. The first one is carbapenem-resistant Acinetobacter baumannii or CRAB. The second one, carbapenem-resistant Pseudomonas aeruginosa or CRPA. The next, ESBL, extended spectrum beta-lactamase producing Enterobacteriaceae or ESBL Enterobacteriaceae. And the last one, carbapenem-resistant Enterobacteriaceae or CRE. So these are common and critical pathogens that need promising antibiotic to fight against them. We also have the pathogen on the high priority list, include vancomycin resistant Enterococcus faecium, MRSA, VISA, and VRSA Staphylococcus aureus, Clarithromycin resistant Helicobacter pylori, fluoroquinolone resistant Campylobacter and Salmonella, and also cephalosporin resistant or fluoroquinolone resistant Escherichia gonorrhea. And this slide shows the global trend of the resistance among Enterobacteriaceae. I will focus on Enterobacteriaceae because these drugs are very emerging problem in my country. And blue bar represent global percent of resistance. Orange bar represent Europe percent of resistance. Dark blue represent North America. Green bar represent Latin America, and pink bar represent Asia Pacific. You can see here that for Asia Pacific country, the percent of ESBL producing Enterobacteriaceae is approximately 17%. Among E. coli, 22%. Among Klebsiella pneumoniae, 10%. And for CRE, it is estimated that 2% of Enterobacteriaceae are resistant to carbapenem in Asia Pacific country. The highest show in Latin America, approximately 5%. And if we focus on MDR pathogen, approximately 8% of E. coli and Klebsiella in Asia Pacific country are resistant, multi-drug resistant. And the problem are strongly high in Europe, as shown in the orange bar. And these are the results from the nearly 200,000 Enterobacteriaceae isolated from 199 medical centers in 42 countries that joined Century program during 1997 to 2016. And this slide shows the depict the carbapenem resistant Enterobacteriaceae rate in Southeast Asia, and Thailand is here as the pale green color, so it means CRE percent in Thailand is approximately 1 to 5%. But for Indonesia, it's around here, right? I'm not sure I point it correctly. The percent of CRE in your country may be around 5 to 10%. So it's a little bit higher than my country. And if we do the subcategory of Enterobacteriaceae is stratified by country. Y-axis represent percent of resistance among all isolate, and X-axis represent each country. You can see here that in Asia Pacific, black bar represent ESBL positive, pale white bar represent carbapenemase positive, metallobeta-lactamase negative, and gray bar represent carbapenemase positive and metallobeta-lactamase positive. You can see here that 20% of Enterobacteriaceae in Asia Pacific are resistant to ESBL producing isolates, and approximately 1 to 2% are carbapenemase producing isolates. The problem is extremely high in my country, in Thailand, when comparing to other countries in Asia Pacific region. And even we know that carbapenem are common in Enterobacteriaceae isolate in our country, we must know the mechanism of resistance, because right now genotype is more important than phenotype. In this slide, we depict red color as the endemic and nationwide distribution. You can see here that KPC is endemic in America and in China. But for India, NDM and OXA are the main cause of carbapenemase in their country. But for Thailand, NDM, OXA are the most common genotypic appearance in our country. WIM and IM are sporadic case in some country, but unfortunately, I don't have any data for Indonesia country. In some hospital or in some country, genotypic test may not be available. So sometimes we can use susceptibility pattern to predict whether they are ESBL, MC, KPC, or metallobeta-lactamase or not. For anyone who do not familiar with the metallobeta-lactamase include NDM and also metallobeta-lactamase that happen in Acinetobacter baumannii. So let's start with the test inhibitor. For ESBL producing gram-negative bacteria, it can be inhibited by chloronic acid. The susceptibility test will show resistance to penicillin, first, second, and third cephalosporin, and almost always resistant to fourth generation cephalosporin. But sometimes they might be dose-dependent, susceptible to fourth generation cephalosporin, and they always susceptible to cefoxitin and carbapenem. For MC producing bacteria, it can be inhibited by boronic acid and cloxacillin. The pattern of susceptibility are exactly the same as ESBL, except it usually susceptible to fourth generation cephalosporin and is always resistant to cefoxitin. But for carbapenemase, you cannot differentiate or distinguish between KPC and MBL by using susceptibility pattern, because both of them are resistant to all beta-lactam antibiotics. So that is the global burden and epidemiology of multi-drug resistant bacteria. I will move to therapeutic option for MDR bacteria. So first, I will focus on O agent colistin, because it is the most active agent for multi-drug resistant gram-negative bacteria in my country. Colistin is an active form of polymyxin. Polymyxin can be grouped into 5 groups: polymyxin A, B, C, D, and E. Active form of polymyxin E is colistin. So we will administer product as a colistin methane sulfonate or CMS, and the CMS will be converted at the kidney to be colistin, which is an active form. The drug is active against many gram-negative pathogens, Enterobacteriaceae, including ESBL producing strain and CRE, Pseudomonas aeruginosa, Acinetobacter baumannii, including MDR strain. However, it is not active against some gram-negative bacteria, including Proteus, Stenomatophila, and Burkholderia cepacia. The most common adverse drug reactions are nephrotoxicity and neurotoxicity. However, in my experience, the neurotoxicity is rare because most of our patients who receive polymyxin are mechanically ventilated. They cannot talk, so they cannot complain about neurotoxicity, but for nephrotoxicity is controlled in the blood test. So more than 50% of our patients who receive polymyxin have problem with kidney. And to confirm the efficacy of colistin, 10 years ago, my colleagues conducted a randomized control trial in Siriraj Hospital. It's a randomized control trial of 93 cases with XDR Acinetobacter baumannii and Pseudomonas aeruginosa. And the study showed that colistin results in 40% mortality and 30% nephrotoxicity, while patients who were treated with non-colistin antibiotic had 80% mortality, which is very extremely high. And as I mentioned before, colistin has a nephrotoxicity, so we need to do the kidney dosage adjustment. We use the very old formula, Cock and Gall formula, to calculate creatinine clearance. Everyone, regardless creatinine clearance, need to receive a loading dose of 300 milligram of colistin or CBA, and then daily dose then adjusted based on creatinine clearance. And for patients who need intermittent hemodialysis or slow intermittent dialysis or CRRT or CAPD, they also need a loading dose, but daily dose need to be adjusted based on the creatinine clearance as well and route of hemodialysis as well. So some colleagues ask me why I love colistin a lot, because we have to choose whether we treat the infection in the patient, but patient will develop acute renal failure later, or choose to let them die with a good kidney function. So I usually choose to treat infection, because we know that if the patient develop acute renal failure, we can consult nephrologist to do dialysis after that. However, nephrologists hate me a lot because I consult them every week. And someone also asks whether we can combine two agents to increase the efficacy of therapy among severe carbapenem-based gram-negative bacterial infection or not. So this slide is the well-known randomized controlled trial study. The nickname of study is AIDA study, AIDA. It's an open-label randomized controlled trial conducted in Europe, published in Lancet ID in 2018. It enrolled patients with documented infection caused by carbapenem-resistant, colistin-susceptible gram-negative bacteria, and the patient need to receive colistin for less than 96 hours. Intervention will divide into two groups: colistin plus meropenem high dose, 2 gram extended infusion in 3 hours every 8 hours. And the comparator was colistin monotherapy. So the slide is this slide shows the result. 70% of patients in this study were infected by carbapenem-resistant Acinetobacter baumannii. 20% infected by other carbapenem-resistant gram-negative bacteria, including CRE. And characteristics were comparable between two groups. Approximately 200 cases in each group. And the study results show that no difference in clinical failure, no difference in 28-day mortality. However, they identify higher incidence of diarrhea among the combination therapy, which can easily explain by C. difficile associated diarrhea or so-called antibiotic associated diarrhea, which quite common among those who receive broad-spectrum antibiotic. However, we cannot explain why we found that lower incidence of kidney injury on day 14 among those who receive combination therapy. However, on day 28, there was no difference in incidence of kidney injury. Based on this study, if you are infected with carbapenem-resistant Acinetobacter baumannii, we do not recommend to use colistin and meropenem combination therapy because there is no clinical benefit at all. Another question is, if you would like to avoid nephrotoxicity of systemic colistin, can be used nebulized colistin, which practically used a lot in my country. You can see here, this is a picture of human airway. And if the particle size is bigger than 10, it will be blocked at the nose level. Is it bigger than 15 micrometers? It will be blocked at the mouth level. For any aerosol or particle size 5 to 10 micrometers, it will stop at first 6th generation of trachea and bronchi. If you want to reach small airways, the particle size must less than 5 micrometers. So any particle, including any drug, that particle size bigger than 5, it may not reach the small alveoli that is the target of antibiotic therapy. So to be sure that particle size that we deliver will reach the small airway that we want, we have to choose the nebulizer appropriately. So right now in the market, there are 3 types of nebulizer: jet nebulizer, ultrasonic nebulizer, and vibrating mesh. For the jet nebulizer, is widely available because it's very cheap. I think everyone ever see this type of nebulizer. Aerosol is generated by Venturi effect on compressed gas. So particle size is vary by flow. If you use the high flow, particle size is small. But if you use the low flow, particle size is big. It's cheap and administration time is long and it depends on flow. If you use the high flow, the administration time is short. Given that the aerosol is generated by use Venturi effect, no heat production at all, so there is no problem of drug instability. After use, they may have some residual volume in the reservoir, and because it's cheap, it can be disposed after use. The second type is ultrasonic. So aerosol is generated by using high frequency piezoelectric crystal. So the particle size is vary by the machine. It's a little bit expensive, but not much, and use a little administration time. But because we use high frequency to generate aerosol, sometimes they produce the heat, and there is the problem of drug instability. And because it's expensive, it needs to be cleaned and reused. And the last one, vibrating mesh. This is the very expensive one. The aerosol generated by high frequency mesh vibration. Size of particle is vary by mesh size. You can choose the particle size. It's very expensive. In my hospital, we have only one machine because this one costs $20,000 per one machine. And there is no drug instability problem because the mechanism is not related to the heat, and because it's expensive, we have to clean and reuse it. However, we have to change the reservoir every time of use, and the reservoir is very expensive as well. In Thailand, the reservoir costs approximately $200 per one reservoir. So only the rich people can use vibrating mesh. So based on these 3 types of nebulizer, vibrating mesh is highly recommended for use as the when we want to use antibiotic in the aerosol form. And to confirm that it can be used colistin nebulized or not, this study includes 20 ventilator associated tracheobronchitis or VAT or VAT case. They use 1 million units of inhaled colistin methane sulfonate which is equal to 40 milligram of colistin-based activity, and the machine that they use is vibrating match. Then they evaluate the colistin concentration in human epithelium lining fluid of human lung. So you can see here the y-axis represent the colistin concentration, and the X bar represent time after administration. So the blue line represent the MIC of bacteria. MIC stand for minimum inhibitory concentration, which mean the smallest concentration, the lowest concentration that can inhibit bacteria. And if you use MIC of 2 microgram per ml, after we administer 1 million unit of inhaled colistin, you can see here that immediately after after administration, the level is okay. At 4 hours is okay, but at 8 hours, only 40% of population had enough. 40% had low concentration. So I think if you would like to use the colistin, inhaled colistin, every 8 hours may not enough. You may need a higher or frequent dose at the moment. And the studies show that for ventilator-associated tracheobronchitis, 80% of patients achieve clinical cure. So that is the like the in vivo study basic side, but we need randomized control trial to confirm the benefit of adjunctive antibiotic. And this slide show my study that conducted at Sirat Hospital 10 years ago. It was an open label randomized control trial of gram-negative bacteria, ventilator-associated pneumonia cases at Sirat. And we use 25 milligram higher dose than previous slide, 75 milligrams CBA colistin-based activity combined with normal saline 4 cc. And administer via jet or ultrasonic every 12 hours. The comparator group is normal saline, and the patient in both group can be treated with systemic antibiotic as appropriate. Based on the study result, mean age of our population was 70 and mean apathy score was 19. 60 to 70% were infected with Acinetobacter baumannii. 30% were infected with Pseudomonas aeruginosa, and 15% were infected with Klebsiella pneumoniae. Based on my study, I could not see any difference in clinical outcome. However, I found better microbiological outcome. P significant 61% versus 38% with P 0.03. And we can see that, you can see here that bronchospasm was more prevalent among adjunctive therapy group, and nephrotoxicity were comparable between two groups. So in my hospital, adjunctive colistin is not recommended. But right now, there is another systemic review and meta-analysis include 7 observational study plus 1 RCT, which is my RCT. So after combined 8 study together, you can see here that in terms of clinical cure, clinical outcome is favor adjunctive colistin combined with IV colistin. So the OR odds ratio of improved clinical outcome with 1.57, which does not include 1. So that means it's statistically significant. However, there was no difference in mortality. Please keep in mind that this meta-analysis include only 7 observational studies and 1 RCT, which I think it is difficult to conclude that aerosolized colistin is beneficial. However, IDSA and ATS guideline recommended to use adjunctive inhaled antibiotic in XDR gram-negative ventilator-associated pneumonia in case if the pathogen susceptible only to aminoglycoside or colistin, because they know if we use high dose of aminoglycoside and colistin, there might be some nephrotoxicity, and also aminoglycoside is not good for lung infection due to PK/PD of aminoglycoside. Also, for the 2019 International Consensus Guideline for Optimal Use of Polymyxin, they recommended adjunctive nebulized polymyxin in XDR gram-negative bacilli HAP or WAP. However, both of these are weak recommendation with very low quality evidence, because only seven observational study and 1 randomized control trial. So that is all about colistin, which is a very old agent. I will talk about 2 novel agents which are available in my country. Before starting with the novel agent, we have to know about the beta-lactamase enzyme first, because right now we can choose the antibiotic based on genotypic activity, genotypic pattern, not phenotypic pattern. Beta-lactamase can be easily categorized into 4 groups, A, B, C, D. Serine base is A, C, and D. Zinc base is B, beta-lactamase. So for A is the example of A beta-lactamase, HHV, cefotaximase, Klebsiella pneumoniae carbapenemase or KPC. C beta-lactamase, for example, MC. OXA48 is considered beta-lactamase class D, and New Delhi methyl-beta-lactamase or NDM is considered beta-lactamase class B. So for the red box, it means drug does not work at all. Green box is okay, so it's active against the pathogen that produce that given enzyme. So you can see here that if the bacteria have beta-lactamase, piperacillin cannot be used at all. But for HHV1, you can use many beta-lactam and beta-lactamase inhibitor. If the bacteria have OXA48, you can use ceftazidime or acetonam or avibactam. But if bacteria have NDM, you must use aztreonam because it's the only agent that active in the class. But right now we have the combination of beta-lactam and beta-lactamase inhibitor. So you can see this slide when we list the combination antibiotic. If the patient infected by ESBL, you can use any drug on the list. But for MC, ceftolozane and tazobactam cannot be used. For KPC, ceftolozane, tazobactam cannot be used. For OXA48, meropenem, valbobactam, imipenem, milibactam cannot be used. For NDM, you can use only acetonam, avibactam, ilavacycline, and cefiderocol. And for Pseudomonas, it also depends on mechanism of resistance. And for Acinetobacter, it also depends on mechanism of resistance as well. So if you focus on this list, the drug that can fight against all types of multi-drug resistant gram-negative is the only cefiderocol, because it's active against all types of resistant mechanism. And also I will focus on ceftazidime avibactam because it's the only new drug that available in Thailand. So we don't have meropenem, valbobactam, or imipenem, milibactam. I heard that some drugs are now available in Singapore and Hong Kong, but it's not available yet in my country. So I will focus on OXA48 and NDM, which are the most common genotypic pattern of CRE in my country and in Southeast Asia. So ceftazidime avibactam is the old beta-lactam but new beta-lactamase inhibitors. The spectrum covers gram-negative, including Pseudomonas aeruginosa, some ESBL strain, and some CRE. that produce OXTA48, but it cannot cover NDM, producing carbapenemate-resistant Enterobacterus E. It cannot cover enterococci, cannot cover MRSA, and cannot cover anaerobe. Someone may believe that beta-lactamase inhibitor can cover anaerobe, but it's not the case of ceftazidime avibactam. It can be used to treat complicated intra-abdominal infection, complicated UTI, hospital-acquired pneumonia, ventilator-associated pneumonia, and also infection from Arabic gram-negative in patients with limited treatment options. The dosage shown here, and it needs renal dose adjustment if creatinine clearance less than 50, but no liver dose adjustment. The most common adverse drug reaction are false positive CUM test and also nausea vomiting. So this slide shows 7 landmark clinical trials of ceftazidime avibactam. The claim 1, 2, and 3 included adults with complicated intra-abdominal infection. They compared ceftazidime avibactam plus metronidazole versus meropenem. For recapture 1 and 2, the study include adults with complicated UTI. They compare ceftazidime avibactam versus doliprane. For the replies, they include adults with ceftazidime-resistant pathogen. They compare ceftazidime avibactam plus metronidazole versus best available therapy. For reproof study, they include adults with nosocomial pneumonia, including ventilator-associated pneumonia. They compare ceftazidime avibactam versus meropenem, and the study can add empiric glyceride and aminoglycoside. Based on these 7 studies, non-inferiority confirmed in all studies. That's why ceftazidime avibactam was approved to use in complicated intra-abdominal infection, complicated UTI, nosocomial pneumonia, and also any infection caused by gram-negative resistant pathogen without limited option of therapy. The next antibiotic is very promising. Cefiderocol is a very new mechanism of development of antibiotic. It's a siderophore cephalosporin. So you can see the structure here. It's a cephalosporin covered by ion molecule. So bacteria, the bacteria do not know this is the antibiotic. So antibiotic will be transport to inner cell via ion-related outer membrane protein, and then they can inhibit the gram-negative cell wall synthesis. The drug is highly stable to carbapenemase. Spectrum is very wide, including multi-drug resistant Acinetobacter baumannii, Pseudomonas aeruginosa, and CRE. Also MC, ESBL, KPC, OXTA48, IM, WIM, and NDM. So it can cover all types of gram-negative bacteria that resist that. That is the global threat defined by WHO. And the dose is 2 grams every 8 hours. The only problem, and is the main problem of cefiderocol, is hepatotoxicity. And also, if you use in the elderly or very sick patient, you may have a problem of fungal infection because ion can promote the growth of fungus. And this slide shows the result from CR study. It's a multinational surveillance study tested nearly 10,000 carbapenem-resistant gram-negative bacteria from North America and Europe. You can see here that nearly 100% of pathogen CR, E. coli, carbapenem-resistant Enterobacterus E, CRPA, CRAB, and also Stenotrophomonas maltophilia. Nearly all of them are susceptible to cefiderocol, while only 70% of CRE in this study were susceptible to colistin, and 90% of Pseudomonas aeruginosa and Acinetobacter baumannii in this study were susceptible to colistin. So the susceptibility result of this drug is very promising. And this is the landmark trial of cefiderocol, two landmark trials, APEC-NP and Credible CR. Both were published in Lancet ID 2020. The first study was conducted in Asia, Europe, and USA. They include patients with gram-negative hospital-acquired pneumonia and ventilator-associated pneumonia. They compare cefiderocol plus linezolid versus meropenem plus linezolid. So in this study, the cefiderocol showed non-inferiority to meropenem and combined with linezolid. However, in this study, the requirement was not include MDR bacteria. Some pathogen are susceptible strain. But for credible CR, they include only carbapenem resistant strain, infection causes by carbapenem resistant strain.
Excuse me, Prof, one more minute. One more minute.
Okay, so the time is out, right?
Actually, the time is up, but if you would like to...
okay, so at the end, I can conclude now. Credible CR confirmed that cefiderocol is non-inferior to best available therapy right now. Okay, and this is my last slide, take-home message. So right now, I think local phenotypic and genotypic epidemiology are important, not as in the past that we focus on phenotypic epidemiology. Right now, we have to rely on genotypic epidemiology. Colistin is an old agent, but it's still active. Ceftazidime avibactam is the first line therapy for some CRE. In the country with the prevalence of KPC and OXA is high. And for the cefiderocol, I think is an ideal agent for most MDR gram-negative pathogen in everywhere. And it is no nephrotoxicity, but with hepatotoxicity. And at the end, I would like to promote the World Antibiotic Awareness Week in 18 to 24 November this year, and would like to insist that prevention is better than cure. So if you can prevent multi-drug resistant bacterial infection, it is easier to find novel or old agent to treat the MDR bacterial infection. Thank you. Okay.
Thank you, Prof. Pino, for the nice presentation. It's time for 5 minutes discussion. First, I'd like to invite Dr. Annette Dearkom. Would you please provide any comments or question to Prof. Pino?
Thank you very much, Achan, for your fruitful information. Actually, I just read about the colistin resistance in my country because now it's almost developed the colistin resistance. And when I try to search that, actually it's already developed in several countries also about the colistin. How about in Thailand, whether this colistin is start to emerge the resistance or it's still effective as like you said in your slide?
Yeah, as shown in my slide is the data from 5 years ago. The colistin resistance is only 5%. It's not high. However, the origin of colistin resistance is occur in pig farm in my country. There is the spread of MCR1 and MCR2. So I think they use a lot of colistin in pig farm because baby pig need to eat colistin every day during their first 30 days of age. I'm not sure if other countries do the same or not, but colistin resistance is very, very common in my country right now. Right now, among CRE, 30% of CRE isolated in my hospital are resistant to colistin. So it's extremely high among CRE. But for the overall E. coli Klebsiella, I think the percent may be less than 1%, but 30% among CRE. So it's also the major problem in my country. Yeah.
Okay, Achan. But because there is a new drugs, right? The cefiderocol that you say that might be it can, but I'm not sure. I think it's not in the market yet.
I think
Thailand
it's right,
and Indonesia.
Not in Thailand and Indonesia, but now it's became available in the United States, in some country in Europe. but we have like some drug to use as a compassionate use right now in my country. Yeah, but
okay.
It's very promising and very impressive with the cefiderocol call. Yeah.
I hope it won't develop the resistance soon also.
I hope so.
And another one, that because we are in the COVID pandemic now and the use of antibiotics is increasing and is more irrational now, do you think it's become will become, or because we didn't use the carbapenem for the, or maybe the higher antibiotics for the COVID-19, or you think it will become a big problem, or actually it's not a big problem?
I think COVID-19 is make people more awareness about prevention control. So right now, previously in my hospital, the prevalence of nosocomial infection is extremely high. But right now, everyone wash hand every time they touch the patient. They wear full protective equipment. So prevalence of drug-resistant bacterial infection is extremely low and very impressive with the COVID-19 outbreak. But for the inappropriate antibiotic therapy, I think it still be there. But among those with COVID-19 patients, I don't think inappropriate antibiotic is the problem, because right now all COVID-19 patients in my hospital were taken care by ID team. So we rarely prescribe antibiotic. Antibiotic usually prescribed after a week or two of hospitalization. So I think due to the COVID-19 outbreak, problem of multi-drug resistant bacteria should be improved, not worsen. So I think it's a good sign. Yeah.
Okay, Acharnka. Thank you very much, Acharn. And I see Dr. Hoirul raise hand.
Yeah, Dr. Hoirul, would you address your question to Prof. Pino?
Yeah, thank you very much, Prof. Pino. It's very wonderful talk. I appreciate it. It is evident that combination colistin and carbapenem for CRGNP, including TPC, Klebsiella pneumonia, carbapenemis, and we know that meropenem is a relatively unstable compound when dissolved. And can we combine it with colistin in a solution for an intravenous administration?
Can you repeat the last sentence one more time?
Can we combine it with colistin? I mean, carbapenem, meropenem, and colistin in a solution for an intravenous administration?
You mean in the same in the same bottle, right?
Yeah, in the same bottle.
Yeah. I don't think I don't think so, because as you know, colistin need to be administered immediately after we dilute the solution, because if you keep the colistin IV fluid now, there is the kind of pro-drug turn to be the active agent. We don't give the colistin to the patient directly, because we would like to avoid nephrotoxicity. We give CMS. CMS will convert to colistin after they reach the kidney of the patient. So if we left the solvent of colistin there, if we left the solvent of CMS there, they will turn to be colistin, and the nephrotoxicity will increase. So the good way to administer colistin is within 30 to 60 minutes to be given. However, for meropenem is very stable. We prefer to use extended infusion. And in my hospital, we always use 3-hour extended infusion. So I don't think in terms of administration, we should combine them together. That is the one strategy. And also, I'm not sure about combine them together, and they may have some interaction that I might not be able to answer the question. Maybe pharmacists will be better to answer this question. but sometimes we use a different bottle but use the same infusion set, and sometimes we can see some precipitation. So we try to avoid to use the same infusion set when we administer both drugs together. I'm not sure I'm answered it correctly.
Thank you. Thank you very much.
Thank you, Prof. Pino. It's a great discussion, but due to limited time, we should end the second session. And I would like the opportunity to express our sincere gratitude to Prof. Pino. Thank you for taking your time.
Okay, thank you. Bye bye.
สวัสดีค่ะอาจารย์ Thank
ส
you
วัสดี
very
ค่ะ
much อาจารย์
สวัสดีค่ะ
It's time for the third session, and it's an honor for me to introduce our next speaker. He has been professor of pharmacoeconomics at University of Groningen and University Medical Center Groningen UMCG, and the Faculty of Economics and Business in Groningen. And I'm very proud to say that he has been adjunct professor at Pharmacology Division, Faculty of Medicine, Unair, since 2018. He is the chair of Global Health Economics, UMCG, and director of UMCG's Research Institute, Chair, Science in Healthy Aging and Healthcare. He leads a team of 100 staff, PhD, and postdoc researchers. He also has extensive lecturing experience in many countries, including teaching our medical students at Universitas Airlangga. He holds of 54 for research publication. He is currently advising the Dutch government on reimbursement of drugs and vaccine, and being a member of UK's Joint Committee of Vaccination and Immunization, and advisor to the All Wales Medicine Group. Therefore, he is the best person who can bring us into deep understanding about health technology assessments of COVID-19 vaccine. Without further ado, please join us in welcoming Professor Dr. Martin J. Postma, PhD.
Thank you. Thank you very much, Professor Norina. Can you hear me well?
Very well, Prof.
That is very good. And thank you very much for the invitation today. And thank you, Dr. Hairul, for the last question in last session. Dr. Hairul, my former PhD, for organizing my lecture this morning. Hi, Rizky. Hi. Shall I share my screen? Is that how we do it in this webinar? Yeah. Okay. Thank you. Thank you. Now it should come. Can you see it?
Very well.
Very well. I'll put it on. I'll put it on.
Choose the view, but it's well seen.
Even better like this, maybe.
Yes, sure.
Okay. Thank you very much for your help. So, as said, Professor Norina, I will discuss the HTA, Health Technology Assessment of COVID-19 vaccines. You already went through my this extensive list. Thank you for that. Good to say that I have hosted PhDs in Groningen in the Netherlands, from a lot from Surabaya and as well as a lot from Bandung. So I'm actually nowadays affiliated to your university as well as to Pajajaran University in your country, and of course in as you said in Groningen as well. I'm obliged always to say that I have a lot of projects going on, and in this context, also with pharmaceutical companies, I should always say when I give any talk anywhere that I receive grants and honoraria from various pharmaceutical companies, inclusive those companies that do develop develop, market, and produce COVID-19 vaccines. So already mentioned, I'm a member of this committee, JCVI, the Joint Committee Vaccination and Immunization. It's a health technology assessment for vaccines. And I must say that last year we had actually no work on other vaccines anymore, and we had meetings twice a week with our committee, online, of course, on the COVID vaccines. So the interest completely on these vaccines, and it has, of course, been incredible. I guess how fast in the world, all over the world, COVID-19 vaccines have been developed. I guess many people would never have expected that within the year after the pandemic was pronounced, within the year, the first COVID vaccines came on the market. I think the UK might have been the first, or at least one of the first countries to officially allow the Oxford AstraZeneca vaccine was approved in the UK in December last year, and the UK was one of the first to start with the vaccination program. And this is the first page of the joint JCVI's announcement at the end of last year on how to proceed with the vaccinations. I will try to keep my talk like within half an hour or so that we have time to discuss on all these issues that I show. From the beginning, I should say that JCVI had a very strong focus on, let's say, prevention of mortality, supporting the National Health Service in the UK in terms of limiting the amount of people going to hospital, and with the NHS related to the social care system. So basically, rather than avoiding or controlling transmission, from the beginning, vaccines were used to limit as much as possible the severe disease and hospital capacity needed for COVID. And the approach taken was, as you can see here, priority listing. It was an age-based approach. There were discussions also, of course, on like bus drivers and teachers, but it has always been said, no, let's keep it simple and straightforward, and we take an age-based approach for the vaccination. And the only basically occupation, profession-related vaccinations were those for working in healthcare. So an age-based approach and healthcare workers first. That was basically JCVI's policy. It's also reflected in this first report, this first report that we wrote. That actually there was also the last point, the bottom point that I highlight here. JCVI does not advise a preference for either vaccine in any specific population. So in the beginning, in Europe, at the moment, still it's a lot of Pfizer and AstraZeneca, and JCVI didn't want to, as least as possible, to prioritize one of the two, to differentiate the two vaccines, basically. And also, therefore, and that's the upper part, the second dose of the Pfizer vaccine may be given 3 to 12 weeks following the first dose, and to align this as much as possible with the AstraZeneca vaccine. It was actually said that both could for sure be given up to 12 weeks following the first dose, whereas Pfizer themselves may argue that it should be a bit earlier. The UK has from the beginning also had a policy that vaccinate as much people, give as much people as possible the first dose, and let's then see how much we still have for the second dose. We also had a statement from another angle, and this might be something that I would like to hear, maybe Hyrule, his opinion. We have this initiative within the Health Technology Assessment, HTAI, Health Technology Assessment International, in the left corner, where we have a strong focus at the moment in the interest group of public health on COVID-19 vaccines, and we are really trying to expand our knowledge, our findings that often in my surroundings come from let's say high-income countries to middle-income countries, let's say. And we would like in this HTAI, for example, very much Indonesian or Southeast Asian representation, which is at the moment still lacking. Just a brief another slide. I only have 2 slides on this, but HTAI, our interest group, is interested in the societal determinants of health, early detection, and preventable comorbidities, and prevention of communicable diseases as certainly one of the 3 strongholds. So this about Health Technology Assessment International. And maybe as a last point, we had a position statement, which is, you know, in COVID-19, everything changes so fast. So this position statement that is a few months ago is of course already outdated again. So we did this position statement is sort of a live document and needs updating every, basically on a continuous basis, and would also require updating with the focus, as said before, to middle-income countries, for example. This is the situation in Europe. Sorry for the Dutch, but it's a quite straightforward slide. It shows the vaccinations in the different countries. You can see the small country of Malta, which is in the Mediterranean Sea, as leading with almost 80%. That is one of the last columns. Almost 80% of the people having their first dose, and fully vaccinated 64%. And you can see in the dots, the colored dots, you can see the types of vaccines that are used, which are actually, as you can see, Pfizer and Moderna is the first 2, and Oxford AstraZeneca, Janssen, which actually I got myself, and Sputnik, and Sinopharm. So if you go down, you can see some countries like Belarus and maybe Bulgaria using some of the, or North Macedonia, Albania using some of the Russian vaccine. Most countries use a lot of the Pfizer, Moderna, and AstraZeneca, and you can see the UK at number 3 in this listing, and the Netherlands is in bold. I think the Netherlands had a difficult start, but now the vaccination program is running very smoothly. So the initial general approach of JCVI, direct protection of those vulnerable first and their carers, avoid differentiation in vaccines, inclusive up to 12 weeks till the second dose for both vaccines that were there initially, and intense monitoring. That was what we did from the beginning of real world data, RWD, real world data, and continuous modeling, both on effectiveness and safety. And I will show you some of the outcomes of the real world data and the and the modeling in some of the coming slides. So if you look at more, so this is really what initially was the approach, and of course things change. So I could say what are some recent considerations of JCVI, and very recent maybe, but I will come back to that at the very end. What about vaccination of children? More recent vaccinations is that we look at benefit risk because of the side effects. You know of the side effects that are discussed on the different vaccines, and what about then the trust in the vaccination program? We have the emergence of new strains like the Delta variant, and the UK did some local outbreak management, in particular in the area of Manchester. The Delta variant emerged, monitoring the results on protection, not only against disease, but what about how good do the vaccines work against infection? First discussions now going on revaccinations, and there is talk about should we do heterologous boosting or homologous boosting, so use a different vaccine preferably, or maybe the same. And I would say from the beginning, limited attention to costs and cost effectiveness, but it is increasing. But with the crisis that we had and still have to a certain extent, nobody really asks about cost effectiveness issues. We basically pay what is being asked. This is some of the modeling done, and this was for basically for the Delta variant months ago, and sorry, quite consistently, the models that are applied in the United Kingdom show around this time, and we might to a certain extent see it, but shows to around this time an increase in the number of, in this case, hospital beds related to the Delta variant, with some specific assumptions that it's 50% more infectious than the British variant that we had before. So this is to illustrate the type of modeling that's ongoing, often done by the LSHTM, as you can see here, London School of Hygiene, Tropical Medicine, as well as by the Warwick University. Two types of models that often indicate the same trends, and these are population dynamic transmission models, and I will show you some example at later in the slides. So then there is a lot going on, and I think there is still conflicting information. This is on the Janssen vaccine, and this one basically shows that that the Janssen vaccine actually here the authors mentioned there are currently four variants, and actually this paper shows that the Janssen vaccine, that was of course never developed for the Delta vaccine, it was a pre-Delta vaccine, that it actually also works against this Delta vaccine. And this is just an example of the type of information that is now coming from everywhere. And some conflicting information is said as well. Sometimes we find that it doesn't protect it well, and sometimes we get reports that it does. A lot of real world data coming in on the side effects. This is the AstraZeneca vaccine, where we had quite some discussion about. This is a picture for in a situation with a relatively low exposure risk, and the issue here is the thrombocytopenia that has been related to the AstraZeneca vaccine. And you can see if we vaccinate starting at the top, 20 to 29-year-old people, actually we avoid 0.8 ICU admissions. However, we have 1.9 cases of thrombocytopenia. So here the risk benefit is actually in favor of not providing the vaccine. It shifts, of course, the older age group we consider, because the risk for COVID increases, and also actually the risk for thrombocytopenia in the data rapidly decreases when we look at older ages. So this was a reason for JCVI to, at a certain moment, despite the fact that the AstraZeneca Oxford vaccine is, of course, a very British vaccine, and a bit of nationalism for sure plays a role. But I think at the moment the AstraZeneca vaccine in the UK is limited to over 40 years of age, exactly because of these risk-benefit analyses. Then we have a real-world data analysis of, let's say, infections. How good do the vaccines work on infections? And this comes from the SIREN study. The SIREN study ran from the beginning that we started with the vaccine campaign in the United Kingdom. This study concludes that the Pfizer vaccine, the BNT, is the Pfizer vaccine, effectively prevents both symptomatic and asymptomatic infections in working age adults. So it actually demonstrates that it works against disease, but not only works against disease, but also against asymptomatic or infection. Then I also mentioned this, another example in the Lancet here from data from the UK and on this issue of revaccinations that we are considering now. Here it is about the side effects of a heterologous boost, heterologous boosting, and as compared to actually homologous boosting. You can see, for example, the CHED. The CHED is the AstraZeneca, and the BNT at the very bottom. You can see the different combinations, and you can see the CHED, which is homologous boosting, or the BNT, which is homologous boosting, and the heterologous boosting, of course, when we combine the two, the CHED BNT or the BNT CHED. And this is on side effects. And in the meantime, also the data came through for effectiveness from this COMCOV study. And already the side effects that is in this slide showed that the heterologous boosting is more reactogenic than the homologous boosting. And when already the thought was that this would probably lead to also higher effectiveness of the heterologous approach as compared to the homologous approach. And indeed, recent data have now confirmed this. So all this data coming through the Joint Committee Vaccination Immunization, and you can imagine it's enormous amounts of data twice a week that we meet on this. Now, is there nothing going on cost effectiveness? Just you know, the cost per QALY or the cost per DALY, per quality adjusted life year, per disability adjusted life year. We are often concerned that new drugs are too expensive, and are they worth their money? Now, there is really not so much, as said, still going on. For example, in the UK, United Kingdom, is a very cost effectiveness driven country, but in COVID, actually, the question has not been asked so far, even once in JCVI, whereas normally for vaccines, it's the first question they ask: is it cost effective? This is a study from the UK. Milton Weinstein is quite famous, a very famous health economist, one of the founding fathers. And they found the graph shows the sensitivity analysis from their analysis. And the base case estimate is the vertical line at around $8,000 US per QALY, per quality adjusted life year. In the UK, 100,000 per QALY is acceptable, so 8,000 per QALY is for sure cost effective. And what the sensitivity analysis shows, and when Hairul Rizki was in Groningen, he did a lot of these kind of sensitivity analyses himself for his studies in diagnostics. And what this sensitivity analysis like this shows is that the estimates are quite robust in the sense that they are always below 100,000 euros, or I should say US dollars per quali. Wat zoek je? Just a second, apologies. Nee, die ligt beneden op tafel. Sorry for the Dutch, it was my wife. So this is the founding father, Milton Weinstein, with a cost effectiveness analysis showing that in the USA, it is very cost effective to do the COVID-19 vaccination. There is this professional organization, ISPOR, ISPOR, International Society of Pharmacoeconomics and Outcomes Research, that have thought about what kind of elements do we need to consider in a cost effectiveness analysis. And again, Hairul applied this flower of value, how it's called, also in his studies. In green, of course, we always look at qualities and costs. In light blue, often we consider production losses, like the lockdowns have immense effects on the economy. And of course, in a cost effectiveness analysis, looking at the vaccines, we should take productivity into account. And then if we go to the dark blue fields, we come into some areas that are generally not yet in cost effectiveness analysis included. But I would say, in particular, also due to COVID, it has opened our eyes that there are various aspects of value beyond the qualities, the net costs and the productivity, and maybe, as also shown here, the adherence improving factors, which are included as well in a cost effectiveness analysis. But think of fear of contagion, just the fact that we are vaccinated and can travel again and don't have the fear to be infected is an important value of the vaccines. The scientific spillovers, the fact that the new technology like mRNA is used and can in the future be used for other fields, the spillover effect. So there is a whole bunch of further values that are currently considered in cost effectiveness analysis. The most important professional organization in our field, the ISPOR, and it also has an annual meeting its annual meeting in Asia. This is a cost effectiveness study that is in I think at least a low middle income country, and it shows some interesting features. As said, not so many cost effectiveness studies being done. But here, if we look at the y-axis, the discounted years of life saved versus the total costs, the total cost on the x-axis, and the numbers per year of life saved shown within the graph. So if we are able to vaccinate 20% of the population, we are faced with a cost of 250 per year of life saved. And you also see some marginal effects here in the sense that, and this could also go the other way around, but it's often not a linear model that you see. So in this case, the higher we get, and if we have already vaccinated many people, and here they go up to, as you see on the right, 70%. If we have already vaccinated many people, the benefits of vaccinating even more in this model decrease. There is, let's say, a decreasing marginal utility of the vaccination. But typical for these cost effectiveness studies is that they are generally not behaving in a linear way. And that is because often dynamic models are used. Dynamic models is something that we investigate a lot also in Groningen. And again, many Indonesian students are involved in these studies. At the moment, actually, one is going into some of the dynamic models for influenza to apply them also to COVID. This wasn't the dynamic models is the core word in this slide, and we applied it to pertussis, but we also apply it to influenza, I said, respiratory syncytial virus, TB, etc., etc. And this slide shows that in light gray, models that have these dynamics in them, which is also called, as you know, herd immunity. Models with herd cost effectiveness analyses with herd immunity. If you want, you can see an increasing trend in the light gray, a bit at the expense of their complement, which is the static cost effectiveness analysis. So an increasing trend in the use of these dynamic models. Here you see a quite clear example of how these models can be used. This was to analyze this paper, vaccinating children against influenza, and maybe not so much for the benefit of themselves, but, and that is what you can see in the graph, if you look in the graph where actually the benefits, and in the graph you see deaths associated quality adjusted life days gained per year per person. And this analysis was on vaccinating children in the United Kingdom. And if you look at the benefits of vaccinating children, they're actually not in the children. They are in the 65 years and over, in the low risk and the high risk groups. So the chronic diseased people, the high risk, as well as the low risk, 65 years and over. So we vaccinate one group, and the benefits are in a completely different group in society. And the only way you can model this is using a dynamic, population dynamic approach. We did the same for the Netherlands. And we looked at the benefits of vaccinating children in the Netherlands. And if you look at, again, at first maybe at the x-axis and focus on the right-hand side, we used a quadrivalent influenza vaccine, the QIV, at the moment in the Netherlands. And let's assume we would vaccinate, we don't do it, but let's assume we would vaccinate the 2 to 17-year-olds. Then the benefits of such a vaccination program would be half the blue part within the group themselves. That is nice, but the other half would be outside the group that is actually vaccinated through herd immunity effects. And this is in particular, so it's below 2 years and over 17 years, but it's in particular, of course, in the 65 and over, where influenza poses the highest risk. And in general, in our analysis, we were quite positive about influenza vaccination, illustrated by this slide, that on the upper hand, we can see the QALYs loss, and on the low hand, we can see the costs of the vaccination program. And if we go from left to right in this graph, we actually see that on the very left, you can see no vaccination, which has the highest QALY loss and the highest costs involved, almost the highest costs involved. And then if we go to the left, it's basically showing situations where we have a better vaccine, like from the trivalent to the quadrivalent, which is also a bit more expensive, obviously. We also could consider here to extend the program to children. So it's getting the program better and more extensive. And basically the trend is that the better and more extensive you make the influenza program, the less QALYs we lose and the lower the costs become. And this, of course, this modeling of influenza has been very beneficial in modeling COVID. And we recently had have developed a COVID model in Groningen. And for one way or the other, we applied it first with a group of colleagues in Turkey, actually already dating back to when one of the PhDs from Bandung was in Groningen. We also did some work with Aulia back then in Turkey. We developed a model susceptible, infectious, recovered, which is often called a SIR model, and people can go from one box to the other, from susceptible through vaccination, get the protection and be as recovered. They can go through an infection and recover. They can die. They can go to intensive care. Not in this slide is that we also included production losses in this analysis. And basically, this is the basic formula that we always apply, which is a differential equation, which basically says that the number of infected people, the number of people that get infected during a certain time period, is depending on the number of infected that we have at that moment in the population as a proportion. Is it 10%? Is it 30%? Is it 50%? That is the IJ divided by NJ. So the higher the number of people infected in the population, the higher the risk that you at that moment attract COVID. And CIJ, the C is what is called the contact matrix. So it describes for any country, and for most countries this information is available, who has contact with who, how intensive are contacts with the people of the same age, or with the people in a workplace, for example. You would probably have a lot of your similar age, but at home it can be grandparents and grandchildren, so the very young and the more old. In table 1, you find some initial starting numbers. Of course, we want to start up the model with numbers of susceptibles and infectious people and already recovered. And then we did our analyses, and we made these kind of heat maps that show the effectiveness of the vaccine on the x-axis and the uptake on the y-axis. And actually, in green, you see that the number of deaths that we still have in the population, very low. And in red, obviously, when we started 30% effectiveness of the vaccine and 30% uptake, which is relatively low. And then we still have a lot of deaths, in this case, in the country like Turkey. But if we would have a 90% effective vaccine and a 90% uptake, we would be all the way up in the green. And we know that effectiveness is more in the area of 90%. We are very positive about that. However, the discussion still is 90% only for severe disease? And does it also apply to infection and transmission? Now, this is our core table, and we have the incremental cost effectiveness ratios. And again, it shows that it's very cost effective to apply the vaccines. We did the discount rate as is mandated by health economic terminology. And from the healthcare perspective, very cost effective, like 511 per quali, very cost effective, 1,000 per quali, if we would assume actually that there is only a limited effectiveness exactly on this transmission. And if we take the societal perspective and include indirect costs, production losses to society, it becomes cost saving. And finally, we made some ISO-ICER curves, which is basically a curve where on any point of that curve, you will find the same incremental cost effectiveness ratio. And you can see here again the curves that are actually not so far apart, again with the x-axis vaccine effectiveness, y-axis vaccine uptake, and the yellow line transmission effectiveness equal to only 50% of disease effectiveness. I think it's what we find in the real world data, that the vaccines are like 90% effective often against hospitalization, but against infection, asymptomatic, it's much lower. And that might be the best guesstimate for how it works against transmission. So around 50% of that 90% against transmission. Now, I will end with some very recent discussions. Of course, we're talking about also in the Netherlands at the moment, how do we implement COVID in the national immunization programs? Is there any seasonality? With influenza, of course, there is this clear seasonality over the world. And many people, some people at the moment would argue because because of the pandemic, you don't see it. It's a pandemic, so much happening. But when things are getting a bit under control, it might well be that we can start to see a seasonal pattern for COVID as well, which I guess, but I'm not an immunologist or a virologist, but I guess could be maybe following a bit the influenza seasonality. Vaccinating 12 to 16 years old, the Pfizer vaccine is now registered. JCVI has so far clearly said we will not do this. The Dutch Health Council has actually recently said, yes, we are going to vaccinate 12 to 16-year-olds as well. And there's some issue on the pricing of COVID vaccines, which is my last slide after I have done this one for the Dutch Health Council. The Gezondheidsraad, the Dutch Health Council, has modeled with their dynamic model, what if we vaccinate from 12 years old, the left panel, and what if we do not vaccinate from 12-year-old, but only from 18 years old. So what about this? This compares basically vaccination of children versus no vaccination of children. Pink is 65% vaccination rate, green 75% and blue 85%. And what you can see is that there is actually quite a difference between the left and the right panels. If we vaccinate children as well, it's about the R number here, the reproduction number, which should be below 1 to have a control of the epidemic. And if we vaccinate children, you can see that we generally keep the R below 1, whereas if we do not in the right panel vaccinate children, the R seems to be often above 1. And then my final slide is on the costing of how should we pay, how should the world pay for corona disease, COVID-19 vaccines, also Indonesia, how should we pay in a situation where these vaccines are quite successful, extremely successful, and maybe some of the pharmaceutical companies think that, I think initially we were surprised by the prices in the sense that they were really not that high. I think in particular, of course, AstraZeneca and Janssen, relatively cheap vaccines of a few euros in Europe. Pfizer more expensive, but certainly compared to other vaccines, still not that expensive. And now this huge success of the vaccination program, if you might say, I think Pfizer, for example, already once mentioned that they want to raise the price, and probably from a cost effectiveness perspective, this would be a fair approach to do that. So how should the world actually is now an upcoming question, deal with pricing of COVID vaccines? So far, nobody has thought about it because there was a pandemic and we needed the vaccines. But now, when we are coming into a bit more, hopefully in half a year or so, a bit more clear control of the pandemic, then we should start to ask, what actually are real prices for these vaccines? So this concludes my talk for today. Thank you for listening, and I'm very happy to answer any question that you have, discussion, and go into discussion with you. Thank you, Professor Nurina, for the opportunity.
Thank you, Prof. Posma, for the nice presentation. First, in the discussion session, I'd like to invite our panelists. Dr. Asra Al-Fawzi, would you please provide any comments or questions to Prof. Posma? Dr. Asra Al-Fawzi? Dr. Abdul Khairul Rizky Purba, would you please?
Yeah, thank you. Thank you very much. Goedemorgen. Goedemorgen, Professor Posma.
Goedemorgen. Goedemorgen. Goedemorgen, Rizky.
How are you?
Yes, doing well. Thank you very much.
It's very nice to see you, even virtually. Thank you very much
Same
for
here.
your excellent and nice talk. It's very, very something new for us to analyze modeling, especially for COVID-19 vaccine. But it's very, very interesting to assess new technology such this vaccine. I have some questions, Professor Posma. Yeah, it's very basic, but if you want to make a model for infectious disease, what parameters that we have to consider? Yeah. In this case, 19.
Yeah, I thank you for the question, Rizky. I always said Rizky. Thank you for the question. I think that what you often see is now let's say basically I think the answer is we need these let's say population dynamic models. We need these for sure more complex models that are often based on these differential equations that I actually briefly showed to catch the dynamics of disease. I showed the example of the UK where we vaccinated children, and we have the main effects primarily in another group in society, and and for COVID it is basically the same. We might want to vaccinate the children against COVID to protect in the end the elderly people that are not vaccinated, or where the vaccine doesn't work so well, or where the vaccine has waned, the immunity has waned. And the only way to actually really analyze this is using these dynamic models. And further on, I think additionally, I would say we need to try to maybe make a model that models influenza, COVID, respiratory syncytial virus, maybe pertussis, pneumonia, all in one, let's say, respiratory model. I would say that would be those are the steps that we now take within a European Union project. Actually.
Thank you very much, Professor Posma. And the other question is about your presentation related to the data among Delta variants. There is a peak in July or August, or might be in January or February for Delta variant transmissions. So from this data, what variables that very, very important to address the time when we should do vaccination?
Because, that is an excellent question. Of course, the answer to your last remark, when should we do the vaccinations? I guess the answer to that is as fast as possible. And that was for sure what the United Kingdom did. The downside of that, of course, is that you might now already see in the UK the very first people being vaccinated, maybe already in December last year. They might show some waning immunity, but in the end, I guess it is the sooner the better. This curve for the UK that I have, that I think you can you see it? Yeah. this this is now one of the discussions that's going on. If we about children vaccination that I addressed at the very end in the United Kingdom, actually this curve, if you analyze it with and without vaccination of children, it doesn't change that much. And interestingly, I showed you at the very end a very similar type of model being used for the Netherlands, how actually in the Netherlands, vaccination of the children, left side, left panel, as compared to no vaccination for the children, right-hand side, makes a huge difference. So actually at the moment we are discussing the Dutch results now also in the JCVI, and we are trying to grasp why these two countries that are so close have so different findings about children vaccination in the context of the Delta variant, for sure, that you describe, Rizky.
Yeah, thank you very much. And for the last, it's about medical tourism. We know that it's very mobile for the people who wants to go abroad, and it's very essential to build dynamic models. And how do we consider the drugs or vaccine is affordable, delivering benefit for people who have tourisms? Yeah, it's about medical tourisms with vaccine, Professor Bosman.
Yeah, which specific group are you considering here, Rizky? I didn't fully get that.
Yeah, it is very dynamic process when we consider vaccine for people who wants to go abroad. So
for travelers, you mean?
It's not prioritized. Maybe the people is younger and not with comorbidity, but I think we need to consider which vaccine is cost effectiveness or not.
Kijk, I think there is a huge, at least if in the Netherlands, there is a huge run on the vaccines for young people so that they are allowed to travel for holidays to Mediterranean Sea. Of course, this goes, you could say, at the expense of vaccinating the older people who really need it. But that is, of course, that could be very different in Indonesia. I don't know about your stock of vaccines and if you have scarcity or whether you have enough. In the Netherlands, there is enough vaccines, and basically, if young people want to travel and get a vaccine, let them do it, because in the end, the more people vaccinated, the lower the R number, as shown in this slide, becomes. So from that perspective, probably also cost effective, Rizky. Yeah.
Sure, it's very nice to investigate. Thank you.
Agreed.
That is my questions. Thank you, Dr. Nurina.
Thank you for the questions.
Thank you, Dr. Khairul, and of course to Prof. Osma. It's a great discussion, but due to time limitation, we should end our third session now. I would like to take opportunity to extend my sincere gratitude to Prof. Osma. Thank you for taking your time. And of course, we look forward to seeing you at our future events. Thank you.
I would be honored, Prof. Nurina. Thank you for your kind words.
Thank you. Before I hand back this event to Dr. Arifa, let me wrap up what we got this afternoon. What we got from Prof. Nazaki, he reminded us how great is our problem in antimicrobial, antiparasitic resistance, especially in our country level, and the fact that our country depends on 90 of pharmaceutical products which are imported products, whereas we are rich in microbial diseases that are potential to be developed as a new promising treatment to address resistance problem. He also emphasized the importance of multilateral collaboration among countries involving academia, industries, government, as well as private institutions. And within academia, solid collaborative are essential, involving clinician, pharmacologist, microbiologist, parasitologist, pharmacist, also expert in biochemistry and biomolecular science, and many others expertises to set up the pipeline of drug development in tackling a resistance problem. Some important messages we got from include the importance of mapping of MDR, XDR, PDR, particularly in our country, employing studies in local typing and genotyping epidemiology. She suggested using old and novel agents. She gives example, the old one listing with possible use of nebulized agent in pulmonary infection caused by particular resistant strain. And also new agent such as ceftazidime combined with avibactam and also cefiderocol. And I don't know how to summarize what has eloquently explained to us, but some message I take is that that the okay in the beginning of pandemic, it seems that we don't really think how much cost should we pay for the vaccine, because of course everybody should be vaccinated as soon as possible. But after we can control this pandemic or it becomes endemic, the study of how the pharmaceutical economy, the cost analysis, and how much people pay for the vaccine should be more important to be conducted. And of course, and also Prof. Osma has described one work he has done with his colleagues in Turkey, that using dynamic compartmental modeling to describe the cost effectiveness of the COVID vaccine, and it is proven to be cost effective, especially from the societal perspective. This all what our discussion today, and thank you for the attention, and thank you especially to our Prof. Martin J. Posma, and also to all of our for taking their valuable times. And it's time for me to hand it back to Dr. Arifah. Thank you for the attention.
Thank you, Dr. Nurina, for your bright conclusion, and thank you for your guidance along these seminars. Especially, I'm very thankful for Prof. Martin J. Posma. We still with you in Indonesia for me again, and thank you for. I am grateful for Prof. Tomoyoshi Nasaki for your nice lecture about how the drug development. And I also thank you for all the participants for your attentions and your kindly to attend these seminars. And I also thank you for all committee and Airlangga webinar conference series team. I think this beautiful afternoon, I will to close this seminar, and I think God bless all of you, and we will have a healthy and prosperous. And I think we would like to invite you to attend our 79th Airlangga webinar conference seminar in July 31, 2021. Thank you, and I will take the time to Airlangga webinar conference teams. Thank you very much. Maybe the Airlangga webinar conference team.
Alhamdulillah lancar ya, Dr. Khairul ya.
Ya, terima kasih Dr. Asro.
Iya, iya wis. Yang lainnya gimana? Mbak Anet masih di sini? Tidak ada ya. Iya wis.
Terima kasih, Dr. Nurina.
Iya wis.
Dr. Anet.
Dr. Nurina ya.
Ini ada Prof.
Prof.
Abdurrahman
Penny juga ya.
Terima kasih Prof. Abdurrahman. Dr. Penny, terima kasih ya. Dr. Anet, terima kasih. Tim AWCS, Rahul dan teman-teman, terima kasih.
Iya wis.
Dok, titip sebentar tanya Dr. Khairul.
Ya.
Ini dari Prof. Zaki bilang kalau filenya terlalu besar kalau mau di-share. Masih perlu PPT-nya atau nggak usah aja ya?
Tadi screenshot apa gimana? Screenshotnya aja ya? Di screenshot ya? Enggak ya?
Nggak enak ya jawabannya gini. I am getting tired of give the same talk few times. The file is too big to send. Nggak enak banget ya.
Iya. Ya monggo, enggak apa-apa sih.
Nggak enak.
Mungkin sebelumnya sering sering nge-share ini ya. Maksudnya sering presentasi ini ya di Indonesia.
Iya, sering. Sering presentasi topik ini. Mungkin kan gede kan gambar-gambarnya banyak.
Oh iya.
Mesti pakai Google Drive,
Iya.
jadi langsung itu. Biasa. Maaf, Bapak saya rewel.